Hello, thank you all for watching this video. I will give you a brief overview of the current status of affairs for the use of pulsed low-intensity focused ultrasound to regulate the immune system. You will probably hear in other presentations that the goal of the focused ultrasound treatments is to initiate a self-sustaining cycle of cancer immunity called the cancer immunity cycle. Focused ultrasound can have a positive impact to initiate and sustain this cycle by killing cancer cells, thus releasing cancer-associated antigens, proteins, and debris, and dangerous signals in the microenvironment, attracting immune effector cells and dendritic cells. There is now a significant amount of evidence for the effects of different types of focused ultrasound in this process. For the purposes of this presentation, I will skip parameters of focused ultrasound designed to produce thermal ablation or histotripsy-like bioeffects. I will go straight to talk about pulsed sequences of focused ultrasound. Pulsed HIFU can be considered distinct from histotripsy in that it uses lower peak negative pressures, typically between 5 and 15 megapascals, and longer pulses. Cohen and colleagues treated entire murine B16 melanoma and 41 breast tumors with 6 megapascals of peak negative pressure, duty cycle of 10%, pulse repetition frequency of 5 Hz, 100 pulses per site, and they assessed the immune cell infiltrates in the tumors, spleen, and tumor draining lymph nodes of these animals at several time points after treatment, like 1, 3, and 5 days. There are many interesting results here. 5 days after treatment, there were significant increases in Th1, regulatory T cells, cytotoxic T cells, macrophages, B cells, and NK cells in the tumor draining lymph nodes of the animals, as you see in these heat maps, where the red suggests an increase in the abundance of these cell subtypes relative to controls. In addition, similar results were seen 3 days after treatment in the tumors, as you see here in this column. Also, there was an absence of significant recruitment of anti-immune cell types like MDSCs. But this study also exemplified some of the challenges that remain outstanding in attempting to dissect the immune response. For example, the temporal effect of the regulation of these biomarkers on the same tumor type was seen. In B16 tumors, significant increases in the abundance of these immune cell subtypes were seen on day 3 after treatment, but not 1 day after treatment or 5 days after treatment. Also, in 41 tumors, a different trend in the regulation of these immune cell subtypes was seen. However, some correlation existed between the two tumor models. For example, an increase in cytotoxic T cells in the 41 and B16 tumors, as you see here, and also an increase in the macrophages in the two tumor types, as you see here. Now, these differences are not unexpected. Understanding the regulation of the immune system, never mind manipulating its activation, is one of the hardest things humankind faces, and all this data have to be seen in this context. So with this background in mind, let's have a look at some examples of the immune effects of low-intensity focused ultrasound. In 2012, Liu and colleagues showed that treatment of CT26 tumors with 1.4 megapascals of peak negative pressure, 100 millisecond long pulses, pulse repetition frequency of 1 Hz and 20 repeats, increased and sustained the abundance of cytotoxic T cells and the ratio of the pro-immune cytotoxic T cells to that of the anti-immune regulatory T cells 3 days and 18 days after treatment when compared to the controls. These results suggest the idea that low-intensity pulse-focused ultrasound can also induce an increase of tumor-infiltrating lymphocytes and change the balance between pro- and anti-immune cells. With a growing interest in applications targeting the blood-brain barrier, 2 studies from 2 different groups showed an inflammatory response at least using some parameters of low-intensity pulse-focused ultrasound when combined with microbubbles. For example, Kovacs and colleagues showed the induction of sterile inflammation associated with upregulation of the nuclear factor kappa beta pathway after treatment of the murine brain with parameters of peak negative pressures of 0.3 megapascals, 10 millisecond bursts due to cycle of 1%, 120 seconds treatments per 9 focal points. Blood-brain barrier disruption and cerebral vasculature vasospasm was observed without causing parenchyma damage or microhemorrhage. In this study, greater infiltration of macrophages was seen to the treated part of the targeted brain relative to the untreated contralateral part of the brain. And this infiltration of macrophages was quantified, as you see here in this slide, with many more macrophages seen in the focused ultrasound treated part of the brain relative to the contralateral part of the brain. As I mentioned earlier, the involvement of the nuclear factor kappa beta pathway in the induction of inflammation in the brain was also seen in the study of McMahon and colleagues. And in addition, what they did in their paper, they compared the upregulation of the proteins associated with the nuclear factor kappa beta pathway to that of the study of Kovacs et al. The result of the two studies for the upregulation of these genes 6 hours after treatment of the brain with low-intensity pulsed focused ultrasound and microbubbles were remarkably similar. As you see in the columns for the genes, you see here the results for the fold change in G expression relative to control in the Kovacs study and in the McMahon study. And the results are very similar. For example, for proteins related to the nuclear factor kappa beta pathway, we see very similar results, very similar increases in the two studies. Now, away from the blood-brain barrier disruption, low-intensity focused ultrasound has been combined with other immunomodulatory agents to produce an effective anti-cancer immune response. Such an example is shown here by the study of Ilovich et al. One of the goals was to increase the infiltration of the tumors by immune cells. The schematic taken by the PNAS paper shows this study's design. Here, anti-PD-1 was injected into the tumor, was injected to the mice, targeted microbubbles and the plasmid encoding interferon beta were mixed and intratumorally injected. Then, low-frequency ultrasound with center frequency of 250 kilohertz, peak negative pressures of 500 kilopascals, burst length of 4 milliseconds, PRF of 30 hertz were applied to the tumor to facilitate interferon beta delivery. And the hypothesis was that once the plasmid was transcribed and translated, interferon beta protein would be secreted, initiating an immune response in the directly treated and the distant tumors. The results of the study corroborated this hypothesis. Not only the tumor cell population was reduced, but also the immune cell population, particularly macrophages, as you see in the graph here on the left-hand side, and also cytotoxic T cells, as you see in the graph on the right-hand side, were increased when compared to controls. Now, finally, low-intensity focused ultrasound can help overcome immunosuppressive tolerance, rendering T cells unresponsive to tumor antigens, which renders T cells unresponsive to tumor antigens, as has been shown by the study of Gua et al. Here, B16 melanoma tumors were exposed to non-ablative low-intensity focused ultrasound at peak negative pressures of 3 megapascals. CD4 positive T cells were isolated from both draining lymph nodes and non-draining cervical lymph nodes. T cells were also obtained from control mice that did not harbor any tumors. As you see in this picture, CD4 positive T cells isolated from the tumor draining lymph nodes produce less IL-2 than cells isolated from the non-draining lymph nodes of the same mice, or from lymph nodes of controlled tumor-free mice when stimulated ex vivo. And these results supported the idea that melanoma tumors induce hypo-responsiveness in a tumor antigen-specific CD4 positive T cells, which translated in a reduced capacity to produce this IL-2 effector cytokine upon restimulation. However, when tumors were treated with low Fu, CD4 positive T cells from the draining lymph nodes of the low Fu treated mice, as you see here, produced more IL-2 compared with the cells obtained from the group of mice bearing untreated tumors. T cells from the corresponding non-draining lymph nodes produced comparable amounts of IL-2 in both treated and untreated animals. These results indicated that low-Fu treatments of the melanoma tumors appear to overcome the hyporesponsive state induced by their tumor microenvironment. Just to summarize, various focused ultrasound modalities, including low-intensity pulse-focused ultrasound, can induce an immune response. The nature of this immune response may be temporal and tumor-dependent. And despite all the evidence, unwanted immune responses may be avoided by carefully selecting low-intensity focused ultrasound parameters. And for that, I will urge you to visit the papers that I have used to create this presentation for more information. So thank you very much for listening, and I hope you enjoy this workshop.